Welcome dear students. In this video we are going to learn about Ras Map Kinase Pathway. In my previous class I have mentioned about the RTK, means receptor tyrosine kinase pathway, and in that video I have mentioned that by the receptor tyrosine kinase pathway, Ras Map Kinase Pathway can also be activated. So what is the significance of Ras Map Kinase Pathway? How this Ras Map Kinase Pathway activates, I will tell you in more details in this class. The first thing you have to understand, that is Ras protein. This is the main central player of this cell signaling. What is Ras protein? Ras is like a G protein. I have mentioned in GPCR signaling pathway, G protein coupled receptor signaling pathway, that there is some G proteins are present. These are called G alpha proteins, which is actually form a trimeric complex called G alpha, beta and gamma. Ras protein also acts like the G protein, but it is not G protein. It is called Gtpage Switch Protein. Why it is called Gtp switch protein, I will tell you. The Ras protein differs from the G alpha protein of the GPCR. And how? The Ras protein is smaller in size than the G alpha protein. Most of the G alpha protein is approximately 300 amino acid. But in Ras proteins, it is actually a very small protein. Approximately 170 amino acids are present. Another important fact is that these Ras proteins don't have any Gtpage domain. So I will tell you what is Gtpage domain, how it works. At first you have to understand the basic function of this Ras protein, that is the Gtpage switch proteins. Why it is called Gtpage switch proteins? Because Ras protein can be present in two forms. One is when it is associated with Gtp, and another is Ras protein when it is associated with Gtp. When is this Ras protein is associated with Gtp, it is off in state. When this Ras protein is associated with Gtp, it is on in state, molecularly on. It can perform its functions. Now, this Ras Gtp can become Ras Gtp, and this Gtp, Ras Gtp, can also become Ras Gtp. How this occurs? There is the two proteins actually perform this function. The first is this reaction from Ras Gtp or off state to on state. The protein is GEF, guanine nucleotide exchange factor. This is a protein that actually helps in the exchange of GDP with GTP. It actually helps in the exchange of the GDP with GTP, and by this way it activates the Ras protein. Another protein called GAP, Gtpage activating protein. I have mentioned about these two proteins in GPCR signaling in more details. What happens? Here you can see when a Ras is associated with GTP, it is on. But when this GTP actually hydrolyzed into GDP, then it cannot perform its function, it is off. So this off is actually performed by, to make it off, GAP protein actually perform its function. Most of the G alpha protein have intrinsic Gtpage activity. You have to understand this. Most of the G alpha proteins, I have mentioned in my GPCR class that the G alpha protein associated with beta and gamma, this is called trimeric complex. Okay, the beta gamma remains separate and alpha remains separate. But this G alpha protein, when attached with GTP, it has its own Gtpage activating domain. That means after a few seconds, approximately 10 seconds, it automatically becomes G alpha GTP to G alpha GDP. But the difference between this G alpha protein and the Ras proteins is that they don't have, this Ras protein don't have the Gtpage domain. So when a Ras protein associate with a GTP, it remains with this GTP longer than the G alpha protein. Approximately it remains 1 minute. In this Ras GTP, that means on state. So GAP protein is very important for the inactivation of this on state Ras. It is very important. So there are two difference, mainly two difference, that is with the G alpha protein, that the G alpha protein is longer, approximately 300 amino acid it is present. In case of Ras, on this 170 amino acid. Another important fact is that Ras protein do not make a trimeric complex, but G alpha proteins make trimeric complex. This G alpha protein has intrinsic Gtpage activating domain, but these Ras protein don't have Gtpage activating domain, so it requires gap protein. One of the example of Ras protein is NF1. NF1. The full name of NF1 is neurofibromatosis. This is a kind of tumor in the neurons. If this protein becomes inactivated, if this gap protein is become inactivated, then what happens? This Ras GTP remains on, and Ras Map Kinase Pathway helps in the proliferation of a cell. So the target of this signaling pathway are cell cycle and cell differentiation, mainly these two. Okay, you have to understand this. So when a Ras protein remains on constitutively, because if there is no gap protein, if there is any mutation in this NF1 protein, then it will not become off instead, remains on instead, and in that case they actually promote cell cycle and tumor occurs. Okay, now how this Ras protein gets activated, I will show you. In my RTK pathway, I have mentioned that the receptor tyrosine kinase receptors always make dimer when bind with its ligand. The ligands are growth factor like IGF-1, insulin-like growth factor. It could be hormone insulin, epidermal growth factor, VEGF means vascular endothelial growth factor, PDGF that means platelet-derived growth factor, NGF that means nerve growth factor. So FGF means fibroblast growth factor. So there are different growth factors are present that can activate this RTK pathway. I have mentioned in my RTK class, so I will not go in details. So I will just summarize this class that when a ligand or growth factor binds with the RTK pathway, the receptor make dimerages, and in the receptor, in the cytosolic domain of the receptor, there is a kinase domain. Both the receptors have the kinase domain. But in inactive state, I have mentioned in RTK class how this kinase domain gets activated. These two receptor kinase domain phosphorylate each other. This side phosphorylate this other and gets activated, and this kinase domain phosphorylate this side, so it gets activated. When this two kinase domain gets activated, they phosphorylate the C-terminal domain of this receptor, particular on tyrosine amino acid, because this is called RTK, means receptor tyrosine kinase. So they phosphorylate on the tyrosine amino acid. Where there is tyrosine amino acid, they get phosphorylated, and this is the beginning of Ras Map Kinase pathway. In this phosphorylated domain or phosphorylated receptor, adapter protein can bind. The adapter protein name is the adapter protein that bind with this phosphorylated state called GRB2. This GRB2 is an adapter protein. This GRB2 binds with this phosphate group by a domain that is called SH2 domain. The GRB2 domain has a SH2 domain that binds with this phosphotyrosine, and this GRB2 protein also have two SH3 domain. Through the SH3 domain, the full form of SH means SRC homology. SRC is a kinase, SRC. In case of Rous sarcoma virus, the SRC protein gets activated and they cause sarcoma. So cancer, one type of cancer. So SRC homology 3, this domain identify different protein structure. This SH2 domain identify phosphotyrosine. The SH2 domain identify phosphotyrosine, but the SH3 domain identify proline amino acid rich region of proteins, and they have two SH3 domain through which it gets associated with another protein. Actually, when there is no ligand, this growth factor, this is the growth factor. When there is no ligand, this GRB2 and the remaining all proteins actually remain in the cytosols. Okay, they are not concentrated, but when this growth factor binds, this receptor dimerization occurs, kinase domain gets activated, then phosphorylation occurs, then GRB2 binds, and then for this GRB2, as these have the SH3 domain, a protein binds with this SH3 domain that is called SOS. This is the very important protein in Ras Map Kinase Pathway. The protein name SOS means, the full form of SOS, son of 7-less. This 7-less is the main player, actually a receptor by which scientists have discovered this Ras Map Kinase pathway. I will show you the experimental pathway, how this Ras Map Kinase pathway actually discovered. So you remember the name 7-less, but the protein is called son of 7-less. The name is very interesting, son of 7-less. This actually the GEF I have mentioned here, GEF. This sauce is the GEF, example of GEF sauce. The example of GAP is NF1. There are so many GAP, there are so many GEF are present. I am giving one example, just sauce and NF1. When this GEF sauce binds with this GRB2, this is the adapter protein. This sauce actually bind with the Ras protein. The sauce bind with the Ras protein, and the Ras is associated with GDP in inactive form. And I have mentioned, when GEF perform its function, then the Ras GDP, off state Ras become on, because it exchanges its GDP with GTP. It helps in this exchange, and actually sauce activates this Ras protein. Now Ras protein activates a cycle, a chain of many different kinase enzyme. I will show you. So you remember this. I am showing here. So you know that Ras protein gets activated. Another information I want to share with you that is very also important, that is this Ras protein is not soluble. It is actually attached with the plasma membrane by a prenylation. In my plasma membrane class, I have mentioned what is prenylation, what is acylation. So this is actually attached with this plasma membrane with prenylations. That means in the C-terminal domain of the Ras, C-terminal domain of the Ras, prenyl group, it could be geranyl or farnesyls. That means it is the multiplication of isopentyl fatty acid. It's not fatty acid, but isopentyl is actually 5 carbon molecules. This is called isopentyl. Now farnesyl is actually 15 carbon molecule, and geranyl actually have 20 carbon. It looks like fatty acid, but it is not fatty acid. This is the like from the isopentyl group also cholesterol also formed. So these are the building block. Isopentyl is the building block which make the farnesyl and geranyl. This could be the farnesyl or geranyl. Okay, these two fatty acid like structure, carbon structure, helps this Ras protein to associate with this plasma membrane. This is called prenylation. Ras protein attached with actually plasma membrane. Okay, attached with the plasma membrane. Now how this Ras protein activate the Ras Map Kinase pathway? This is important. Ras protein can activate a chain of kinase, like Ras actually activate Ras GTP, activate RAF, a serine-threonine kinase enzyme. This is RAF is an enzyme, but this RAF enzyme cannot phosphorylate tyrosine. Okay, now RAF enzyme activate another enzyme called MEK. This is also a kinase, K for kinase. So this MEK is the dual specific. This is the dual specific kinase. That means it can phosphorylate serine-threonine, also tyrosine, both these amino acids MEK. Now the MEK phosphorylate another kinase called MAPK, MAP kinase. That is the ultimate kinase, MAP kinase. This is also a serine-threonine kinase. Serine-threonine kinase not can phosphorylate tyrosine. So you have to remember this. Cannot phosphorylate tyrosine. And ultimately MAP directly phosphorylate a transcription factor called TCF. Directly phosphorylate transcription factor called TCF. Actually MAP, when gets activated, it enters into the nucleus and phosphorylate TCF, a transcription factor that is present in the DNA, attached in the DNA. When this TCF gets phosphorylated, another thing, this MAP kinase actually phosphorylate a cytosolic kinase also, that is called P90 RSK. P90 RSK. P90 RSK. This is another kinase. MAP kinase activate this enzyme that is present in the cytosol. Then this enzyme can enter into the nucleus and can phosphorylate another protein called SRF, serum response factor. SRF. It phosphorylates the SRF, and these two SRF associate with this TCF and helps in the transcription of some early response genes. Early response genes, what is early response genes? When any growth factor binds with any receptor, RTK receptor, then many genes actually get activated, transcribed. And the reason of this transcription is for cell cycle. They promote cell cycle. Example, I want to give you one example, one gene that is actually transcribed, that is called CFOS. CFOS. Okay, and the region where this two transcription factor binds is called SRE, serum response element. Why is called serum? Because many growth factors are present in the serum, our blood serum. So it is called serum response element. This site, this location, this promoter site is called serum response element. And the gene is CFOS. CFOS associate with C-Jun, another transcription factor, and make AP1, a complex. This is the whole story. Now I will show you how these occur, how RAF actually gets activated, because there is a molecular mechanism. You must note down this, what I have draw here. Okay, so how RAF gets activated? The thing is, when the cell is unstimulated in condition, that means there is no growth factor in the cell surrounding of the cell, when there is no activation of this RTK pathway, then this RAF, let's assume this is the cytosol and this is the nucleus. Let's suppose this is the nucleus, this is the cytosol. So when there is no phosphorylation occur or any activation occur of the RTK receptor, then the RAF protein actually remains in the, this is the RAF protein, remains in the cytosol. But the RAF protein is actually phosphorylated in conditions. In that situations, this RAF protein is sequestered. Sequestered means it is actually captured by a protein called 1433. This is actually domain. This is actually 1433 is a domain which helps in the sequestration of any protein. They bind with phosphoserine. Phosphoserine. This molecule binds with phosphoserine. So this phosphorylation actually block the activity of RAF. But the RAF have a N-terminal regulatory domain. This is the N-terminal regulatory domain. I have mentioned when any growth factor binds with the receptor get phosphorylated, then GRB2 bind, then SOS bind, then RAS gets activated. Now this RAS protein actually attach with this RAF. RAF actually, when RAS protein gets activated, that means it is associated with GTP, then RAF bind with this RAS GTP. And that means in this way, this RAF protein actually associate with the RAS. So RAS associate with the RAF protein. Also the 1433 protein also present, but this RAS GTP, when actually hydrolyzed, this GTP into GDP, Okay, at first it was Ras GTP. When it associates with this complex RAF and 1433, the Ras GTP actually hydrolyzed into Ras GTP, and in that situation this Ras protein are no longer bind with this RAF protein, and the RAF protein also dissociates this 1433 protein, and the Ras become active. Ras is a kinase, so it also have a activation loop. I have mentioned the role of activation loop in RTK pathway in more details. You must watch this RTK video, because then you cannot understand how this RAP or other all these kinase actually gets activated. Because in that class I have mentioned there is the activation loop that actually masks. Let's suppose this is the kinase domain of a protein, red sign, but the activation loop, suppose this is the activation loop, actually mask this kinase domain like this. When this activation loop get phosphorylated, it's by this activation or interaction with this Ras protein, then the phosphorylated this activation loop can no longer mask this kinase domain, so it actually changes conformations so that the kinase domain becomes active. Okay, the RAF protein is a serine-threonine kinase. It can phosphorylate the activation loop of this MEK. When the MEK activation loop gets phosphorylated, it becomes active. This is a chain reactions. This become active. When MEK get activated, they also phosphorylate the activation loop of this MAPK kinase. Okay, you have to understand this. The activation loop is actually gets phosphorylated. Okay, and then it cannot hide. Actually, activation loop hide the. Let's suppose I am giving you examples. Suppose this is the activation loop, and here is the site of the kinase domain. Let's suppose this is the kinase domain I have mentioned in red, but the activation loop actually prevent or mask this kinase site. But when this activation loop get phosphorylated, it changes conformation so that it cannot mask the or hide the kinase domain. So now the structure will become like this. Let's suppose now this kinase domain is open. It changes its conformations. This activation loop changes conformation. And when MAP kinase get activated by the phosphorylation of this activation loop, this MAP kinase form dimer. You have to understand and you have to remember this. This MAP kinase form dimer. This MAP kinase can directly activate another kinase called P90RSK, and that actually phosphorylate, actually enter into the nucleus and phosphorylate SRF, serum response factor. 2 SRF bind with TCF, that is another transcription factor, ternary complex factor. This MAP kinase can directly enter into the nucleus, and by this way it activates the early response genes, and in that situations many early response genes get activated, and that actually regulate cell cycle. So in a way you can understand that if there is any problem or mutation occur, if the Ras protein gets activated constitutively, then there is a problem, then the cell will proliferate much more than the normal condition and can cause cancer. Here I want to mention another fact about the Ras protein.